Carbohydrates, lipids, proteins, and minerals are major chemical components that can be found in the seed. In this lesson, we will look at the properties of the major reserves that are stored in the seeds, starting off with the carbohydrates. The carbohydrates, lipids, proteins, vitamins, and minerals in the seeds are present in various proportions depending on the plant species. All of these components are used by the embryo when it starts germinating and even until the early stages of seedling establishment. These chemical components are found in all the basic structures of the seed, including the endosperm, cotyledon, seed coat, and the various tissues that make up the embryo. This slide gives us a general overview of the various chemical components that can be found in a wheat seed. This is a cross-section of a wheat seed showing the endosperm, the structure right here, as well as the embryo, which is labeled as germ in this picture. If you look closely in the composition of the endosperm right here, you can see that it is mostly composed of carbohydrates in the form of cellulose, hemicellulose, and starch. Aside from carbohydrates, there are also minute amounts of proteins, vitamin B, and iron in the endosperm. Now, if you shift your attention to the embryo or germ, you can see that it also contains carbohydrates in the form of cellulose and fructans. And aside from carbohydrates, the embryo also contains fats or lipids, vitamins B and E, as well as minerals. There are many other chemical components found in other tissues that make up the whole seed, including the bran and the alurone layer. In the right, we have a tabulated form of the chemical composition of a corn seed or kernel. The endosperm of corn, like the endosperm of wheat, is composed largely of carbohydrates in the form of starch. We can see here that the endosperm is composed of 88% starch. But it also contains small amounts of oil and also protein. Now, if we look at the composition of the embryo, we can see that its major component is oil or lipids. Starch is also present, but at a significantly smaller amount compared to that present in the endosperm. The proportion of each of the major chemical components of seeds varies depending on the plant species. Most cereals like barley, corn, oats, rye, and wheat are going to have higher proportions of carbohydrates, as you can see here. In this table, the range of carbohydrate content for cereals is 66 to 80%, and this is mostly found in the endosperm. Plants that are considered as oilseed crops will have higher proportions of oils or lipids, and we have here some good examples in peanuts, pine, palm, and castor bean. As you can see, these seeds contain higher proportions of oil than carbohydrates. The oil content in the seeds of these species range from 48 to 64% and can be found either in the cotyledon or in the endosperm. Carbohydrates constitute one of the major chemical components of seeds. Carbohydrates can come in the form of stored food reserves or non-food reserves. Carbohydrates that are stored as food reserves will provide food for the embryo during germination. Starch and hemicelluloses are two examples of carbohydrates that are stored as food reserve. Starch is the main stored carbohydrate in seeds, especially in cereals like wheat, corn, and rice. They are usually found in the endosperm or cotyledons. Most starches are metabolically inactive until they are needed during germination, and a good example of this can be observed in barley. This slide should look familiar to you. In this slide, we have a cross-section of a barley seed with the starchy endosperm surrounded by the alurone layer. The starchy endosperm is made up of dead or metabolically inactive cells that store starch and proteins. From our lecture on seed morphology and anatomy, we've already established that the endosperm is the source of food for a growing embryo during germination. In this example, how do you think will the embryo use the carbohydrate if the cells storing the starch and proteins are metabolically inactive? Now, the starchy endosperm, as you already know, is surrounded by the alurone layer. And this is just an enlarged portion of the alurone layer. In a mature seed, This alluron layer is alive or metabolically active. During germination, the alluron layer functions as a digestive tissue that synthesizes and secretes large quantities of enzymes that will mobilize food reserves from the starchy endosperm to the growing embryo. In seeds, starch is stored in two related forms, the amylose and the amylopectin. Amylose and amylopectin are both made up of glucose units, but they differ in the amount and in the way they are packaged in the seeds. In these illustrations here and here, each pentagon structure is a glucose molecule. In amylose, these glucose molecules are arranged in a linear fashion, and each amylose molecule is going to be composed of 300 to 3,000 glucose units. This linear arrangement of glucose molecules makes your amylose look like single threads like this. The amylopectin, on the other hand, is not linear but branched. As you can see here, look at the branching that the glucose units are making in this structure. Compared to amylose, the amylopectin is composed of more glucose units, which can go as high as 10,000. Starch in seeds contain more amylopectin than amylose. Amylopectin makes up 75 to 80% of starch, while amylose only make up 20 to 25% of starch. Amylopectin is water-soluble and forms a gel with the addition of water. Amylose do not form a gel with the addition of water because it is insoluble in water. Enzymes like alpha and beta amylase can completely break down amylose, while only 50% of amylopectin can be broken down by the same enzymes. We will discuss why this is so in succeeding lectures. Finally, amylose gives a dark blue-black color when stained with iodine, while amylopectin will give you a reddish-brown color. Aside from starch, hemicellulose is another major form of storage carbohydrates, or carbohydrates that are used by the embryo as food during germination. Hemicelluloses are reserved food materials that are usually found in the thickened tertiary layers of cell walls of endosperms or cotyledons. They can come in various forms such as silans, mannans, galactans, xyloglucans, and glucomannans. Hemicelluloses are characteristic of seeds of many palm species, including date palm and ivory nut palm. This slide shows hemicellulose in the form of Mannan cells that make up the endosperm of a date palm. The endosperm cells were stained with toluidine blue O. This dye only stains proteins but not cell wall mannans. So this structure that you see here encircled in red is one cell in the endosperm. You can see an unstained portion of the cell right here. This is the mannan. And then inside we have a portion stained in blue. This is the protein. Aside from the storage carbohydrates, seeds also have non-storage carbohydrates in the form of pectins and mucilages. Pectins and mucilages usually have a different function aside from providing food for the embryo during germination. Pectins are structural heteropolysaccharides that are usually found in cell walls and middle lamella. Pectins can be used in jam making as a gelling agent. Mucilages, on the other hand, are carbohydrates composed of polyuronides and galacturonides that chemically resemble pectic compounds and hemicelluloses. They usually envelope seed coats and help in water absorption during water imbibition of seeds. They are physically similar to the gums found in barks and stems of many plants. You should also be familiar with this slide. This slide demonstrates the presence of mucilage in Arabidopsis seeds. The one on the left is an Arabidopsis seed stained with ruthenium red. The outer cloudy layer and the darker staining layer close to the seed is the mucilage. To the right, we have a mutant seed without the mucilage. That's why you don't see any light or dark colored staining surrounding the seed. In summary, seed reserves are stored in the form of carbohydrates, proteins, lipids, and minerals. These reserves are found in the endosperm, cotyledon, and various tissues that make up the embryo. Carbohydrates are the major storage reserve in the seeds. The major forms of storage carbohydrates are starch and hemicelluloses. The starch can be in the form of amylose or amylopectin. Amylose and amylopectin differ in the number of glucose units they contain and in how the glucose units are arranged. The non-storage form of carbohydrates includes pectins and mucilages.